Well, as is usually the suspicion, the young scientist gets left for the absolute last talk of the meeting. And Stuart, why don't you come on up then, and we'll save a little time when we get reorganized. Not only do we give you the last talk, but then we say, we're running a little behind. Sorry. The Raymond Lindeman Award is to recognize an outstanding paper written by a young scientist. And no pressure or anything, but this room is full of fabulous scientists, several of them right down here in front, who've been awardees of the Lindeman Award. A spectacular honor, I think, for a young scientist, and much deserved. Stuart is recognized this year with ASLO's Raymond Lindeman Award. He was nominated by Jay Lennon for this very fine paper in PNAS, discussing how dormancy contributes to the maintenance of microbial diversity. I don't want to steal any of Stuart's time, but the citation is very important. Dormancy is a bed hedging strategy used by various organisms to overcome unfavorable environmental conditions by entering a reversible state of low metabolic activity. He's going to tell you all about this. Maybe I should just cut to the chase. His keen intellect, that's the part we like, creative ability and interdisciplinary training make him a very worthy recipient of this year's Raymond L. Lindeman Award. And congratulations, Stuart. Thank
you very much. Thank you, John, and good morning to everybody. I'd like to start with a few thank yous. Thanks to the award committee for the recognition and allowing me to speak this morning. Thank you to the organizing committee and our hosts for a wonderful, wonderful week. I've learned an amazing amount in a short period of time. I'd also like to ask the students and young investigators in the crowd to thank the leadership of ASLO and a bunch of senior members of ASLO who put in a whole lot of time for a whole lot of events this week to help us out. So let's give them a round of applause, please. And then last, I'd like to thank Jay, who's a co-author in this work and also nominated me. As John alluded to, it's sort of daunting to be recognized for this award as it's named after Raymond Lindeman and all the very significant contributions he made as a young investigator and over such a short period of time. It's also great, as someone who thinks a lot about microbial systems and bacteria, to be recognized for this award named after Lindeman, because he was sort of a early thinker and recognized the importance and the centrality of bacteria in aquatic ecosystems and how important they can be. Of course, because of the era that he worked in, he really had he didn't have the means to recognize or respect the immense diversity that he captures in that small circle that he calls bacteria, both genetic and taxonomic diversity. He also didn't yet have the ability to comprehend the complex and the complex interactions and the diversity of those interactions between the bacteria and that stuff he calls ooze. Today I'll tell you a little bit about a paper where we explored one mechanism that might explain some of the ways to maintain that immense diversity and which may mediate some of the functional properties of this group of organisms that's important in aquatic environments. We all know that environments, aquatic, terrestrial, any sort of environment are very dynamic through time. And as Nelson mentioned earlier, organisms have to adapt and evolve strategies to deal with this variability. And one ecophysiological trait or mechanism by which organisms can confront this diversity is dormancy. And there are many examples that have been studied for a very long time of dormancy in macroorganisms, such as the seasonal hibernation of mammals to avoid the cold, low-resource winter. Annual plants or plants in general producing seeds that can last for decades and centuries. And an aquatic example here, the Ephippia formed by Daphnia and resting stages of other zooplankton that can, as Nelson showed us, last for decades as well. And of course, these have implications for both the long-term individual fitness of an organism, but also can enable coexistence, has been shown over and over by Peter Cheston and others with storage effect theory and things of the like. Also, as John mentioned, there are many great researchers who have previously received the Lindeman Award, including Carla Caceres, who worked on some of this storage effect theory in a zooplankton system. Of course, we shouldn't leave bacteria and microbes out of the party. There is a whole lot of evidence that microorganisms are able to participate in this mechanism. They can do dormancy. There are some fairly well understood and detailed examples of this from the laboratory and medical field. So one example is tuberculosis. Mycobacterium tuberculosis that causes this disease. It can lie latent in a human for a long period of time and then emerge as a full-blown disease. We also have some evidence from natural systems. So in this image, it's an epifluorescence image. We have a green fluorescent DNA stain, which stains all bacteria that are present in the sample green. And then there's also another reagent added that when in the presence of an active electron transport chain, it forms red fluorescence. So each of these red little dots is an active bacteria, where you see a green dot that's an inactive bacteria. So we know in the ocean and in lakes for decades that some fraction of the total bacterial community is active, and there's a large portion that is inactive. Isabel Reich also showed us a very charismatic example of microbial dormancy yesterday with Darwin's dust and the organisms that were able to be grown after a long-term resting stage in this vial. So with my remaining time, I'm going to quickly buzz through the paper that the award is for and try to answer this question of how dormancy may impact the maintenance of microbial diversity in lakes and also community composition in sort of a lake region context. So because of that strong foundation of theory that's been developed by Cheston and others, we wanted to start at that place too, and we've adapted some of that theory to apply to microorganisms. So you can think of a community of bacteria. Each population can each individual can join one fraction of that population, either an active state or a dormant state. The active state is able to reproduce and is subject to mortality. Dormant state is also subject to mortality, but often that's a lower rate of mortality. And then the individual can shift in and out of that, in between the dormant and active state. Because I'm going to show you some empirical data as well in a sort of lake region context, we considered lake region or a collection of patches. And we also wanted to think a little bit about environmental cues at a couple scales. So the governing equations of the movement between active and dormant states in each population are driven by two environmental factors, one at a broad regional scale and one at a local scale. So each organism is exactly the same except for its sort of regional and local niche or response curve. And so in different manifestations of the model or simulations, we considered a strong regional cue, which is sort of going to homogenize the region, or a very heterogeneous landscape with strong local environmental cues. And then we can manipulate the strength of dormancy or the ability of microorganisms to go dormant by changing this mortality rate of the dormant pool. As we might expect from all of the historical theory developed in macro communities, we see a strong effect on local richness of dormancy. So increasing dormancy or a decrease in that mortality rate from the dormant pool along this axis, and we see a strong effect, less effect on alpha diversity by the strength of this regional cue. Because of the way that we can collect our empirical results, we discriminate between an active and then a total pool of model community, of the populations in the model community, rather than an active and a dormant pool. That has to do with how we measure the system. So if we run a model simulation where we look at community composition in this clustering dendrogram where we have low dormancy and a heterogeneous landscape, we see close coupling of the active and the total pool in each location, each community. And basically, things that are there are active. However, if we increase the ability of organisms to go dormant and we impose a strong regional cue, we can get a stark distinction between the active pool and the total pool. And I like to think of this as sort of a mother spaceship beaming down a signal that wakes up the same populations within each of the local communities. So with a little bit of expectation in hand, we set out to compare some empirical results to the results of that theory exercise. We surveyed 8 lakes near Kellogg Biological Station in Michigan. We looked at the environment using regular limnological techniques, measuring total phosphorus, pH, things of that sort. And then we have to use molecular techniques to dig into that diversity that I mentioned earlier in the microbial community. And we used a fairly simple technique, terminal restriction fragment length polymorphism, to look at bacteria and eukaryotic composition. But we did this with DNA and RNA as a way to get at the active and total community composition. RNA being active, DNA being anything that's present. A quick run through of how we sampled that microbial community. So we use a marker gene that's a ribosomal RNA marker, 16S or 18S, depending on bacteria eukaryotes. We would expect a very active cell to have a large number of ribosomes, but a constant chromosome with this marker gene being transcribed into RNA. So an active bacteria would have a whole lot of ribosomes and have a high RNA to DNA ratio. Whereas an inactive bacteria would not have a whole lot of ribosomes, it's not making a whole lot of proteins, and it would have a relatively low RNA to DNA ratio based on this marker. So with that tool in hand, we went out and sampled, as I said, eukaryotes and prokaryotes in that 8-lake region, and we see an interdispersion of the active and the total community based on our metric when we look at eukaryotes, suggesting that the things that are there are active, consistent with a model simulation with fairly low dormancy. When we look at the bacteria, we see a really strong distinction that, again, is reminiscent of that model iteration where we have fairly high dormancy and a strong regional Q, suggesting that dormancy may be more important, as we've measured it in bacteria, than eukaryotes in lakes. We can also tally up the total number of populations that are there based on DNA and the proportion that are active based on the RNA, and we see another contrast between the eukaryotes and prokaryotes. Eukaryotes are low in their proportion of dormant populations and consistent across this index of resource availability in the lakes. Bacteria we see can be much higher, but they respond to that resource availability, which makes some sense to us. If there's high resource availability, there's no point in being dormant. There's less stress. So about 7 years ago, I'll say, we gained some new techniques and were able to sample microbial communities in a deeper way, and we, just like in many other communities, plant communities, animal communities, we see this log normal distribution, a rank abundance curve, but some argued that the tail is much, much longer in bacteria. And so we wanted to explore how dormancy, the way that we're measuring, may interact with this log normal distribution and the high prevalence of rare taxa. We had a couple different hypotheses about how that could work out. This is sort of a null hypothesis where dormancy is not important, and based on some previous results I've shown you that this may not apply much, but this is basically if you're there, you're active, and the RNA and DNA measures would correlate strongly. We may have that the abundant things are the active things, and then this long tail is just a deep seed bank that just hangs out there. Or we might have that the abundant things are abundant because they were active yesterday, and now something new is climbing up the food chain or up the corporate ladder, and they are highly active when rare. When we sample that for 2 lakes, we see exactly that third case, that the rare things are disproportionately active based on this logistic regression, and the abundant things tend to be more dormant or disproportionately dormant based on their abundance. And so to us, that suggests that the rank abundance curve is maybe more dynamic than we sometimes give it credit for. Things that are abundant were likely responding to previous environmental conditions that are now on their way out of the system, or at least down this rank abundance curve, while other things may be climbing up. The ability for a population to go dormant allows it to reduce its mortality rate and avoid extinction, and it creates this seed bank that is able to respond, both maintaining diversity and some functional redundancy in the system. With that, I'll leave you with some summary points, and I'm happy to take any questions. So thank you for your attention.
We would have time for one short question. Since Stuart stayed on time. I'm seeing fatigued faces,
Yes.
Stuart, but thank you so
Enjoy
much.
Kyoto.